Assalamu alaikum wa rahmatullahi wa barakatuh, everyone. Good afternoon. I'm delighted to be one of the speakers at this distinguished forum in the region. And now it gives me great pleasure to welcome you all to the second session where the module is titled New Insights into Rare Diseases. And we will take you into a journey this afternoon to explore rare diseases with our distinguished speakers. And we will start with Dr. Valérie Delague. My French is not that good, Valérie, so she's a research director at INSERM, where she is currently the leader of the group Genetics and Pathophysiology of Inherited Peripheral Neuropathies in the team of Translational Neuromyology. She's also the scientific head of the Genomics and Bioinformatics Facility at Marseille Medical Genetics in France. She received her PhD in biology from Université Montpellier in France again. Valérie will tell us today the story of Charcot-Marie-Tooth disease type 4H from gene discovery to treatment. Valérie, the floor is yours.
Hello, everyone. I'm really glad to be here today, although I'm so sorry I could not come to Dubai. I would have liked to visit the city. I have never been there. So as Mariam said, I'm going to share my screen first. Is it okay for everyone? You can see my screen?
Yes, we see.
Okay.
Yeah,
so I just want to add that I spent many years in Lebanon, where I was working with Dr. Megarbane, for I think you are a certain number of you who know him, where we did great genetics there. And that's why now I'm focused most of my research on studying the genetics of Charcot-Marie-Tooth disease on large consanguineous families from the Middle East and the Mediterranean area. So today I'm going to speak about Charcot-Marie-Tooth disease type H, to tell you the story of this Charcot-Marie-Tooth subtype. That we started from the families who had no diagnosis, no idea of their disease, of the gene which was causing the disease. And now maybe we hope we are close to a treatment. So just to begin with, I will do a reminder about what is Charcot-Marie-Tooth disease and what are inherited peripheral neuropathies. So yes, okay, so inherited peripheral neuropathies, it's a large group of disease, a hereditary disease which affect the peripheral nervous system. So it's here in red. So the peripheral nervous system, it includes the spinal nerves and also the 12 pairs of cranial nerve. And the role of this peripheral nervous system is to convey information from the brain to the muscles for voluntary movements, and also to carry information from the periphery to the brain through sensory nerves. Among this large group of inherited peripheral neuropathies, there is the most prevalent subgroup is Charcot-Marie-Tooth disease, also called hereditary motor and sensory neuropathies, which affects both motor and sensory components of nerves. The disease has been first described in 1886, so quite a long time away, by three neurologists, two neurologist from France, Jean-Martin Charcot, who is a famous French neurologist who discovered a large number of neurological disease, and his pupils, Pierre-Marie, and one British doctor, Howard Tooth. They gave their name to the disease. And they described this disease, as you can see here, as a progressive distal muscular weakness, as characterized, by progressive distal muscular weakness and atrophy, which begins at the lower limbs, at the distal extremity, and which slowly progresses to the upper limbs. It is quite, it's a rare disease, but it's quite prevalent, with a prevalence of 1 out of 2,500. And it is also characterized by typical feet and hand deformations, and sometimes also other skeletal deformations such as scoliosis, as you can see here. We usually subdivide this group of disease, Charcot-Marie-Tooth disease, because again, it's highly heterogeneous and you will have different subtypes of the disease. We have demyelinating Charcot-Marie-Tooth disease and axonal Charcot-Marie-Tooth disease. Demyelinating CMT primarily affect the Schwann cells, which are the cells constituting the myelin in the peripheral nervous system, as you can see here. And the other group is axonal CMT, which will affect the axons from neurons, so either sensory neurons or motor neurons, which come out of the spinal cord here. And I would like you to know that nerves, peripheral nerves, are called mixed nerves, meaning they carry motor information and sensory information through motor or sensory fibers. It is clinically impossible to distinguish demyelinating and axonal CMT, but to know whether it's one or the other, we can do electrophysiology. And the demyelinating subtypes are characterized by diminished nerve conduction velocities, while axonal CMT are characterized by maintained nerve conduction velocities, but decreased action potential amplitudes. The group of Charcot-Marie-Tooth disease is a model of heterogeneity. It's a very heterogeneous genetically, with around 100 genes described to date, as you can see here. We have mutation on mitochondrial DNA, which is a problem, of course, as you all know, for diagnosis, because we have to deal with many variants in many genes, and which also underlies complex pathomechanisms. Indeed, all the genes which are mutated in Charcot-Marie-Tooth disease are important for the biology of normal peripheral nerve system, and they might be important either for Schwann cell here in it's not red, I forgot the color, my English is so bad, I'm sorry. Here, the Schwann cells. For example, you will have genes encoding proteins that are important for the structure of myelin, such as PMP22 and MPZ. Or you will have genes which do strictly have a role in the neurons here in blue. It might be motor neuron or sensory neurons, such as, for example, kinesin and dynein, which are involved in axonal transport. There are also genes which encode proteins having a role both in Schwann cell and in the neurons. And to complexify a little bit more the situation, which is already very complex, there is a very tight relationship between Schwann cell and axons, which do have to act together to myelinate and to provide the correct transmission of the nerve influx through the axon. So I will speak today about one specific CMT subtype, which is CMT4H, that we have described. It was done in collaboration with the team of Nicolas Lévy, who is my actual boss, when we began this study back in 2002, so it's a very long story. I was still working in Lebanon at Professor Megarwane's lab, and we had this very large family, as you can see here, and you know very well, such large consanguineous families with a high number of affected individuals, and also one other Algerian family, also consanguineous. All patients share the same clinical features. So they are affected with autosomal recessive demyelinating neuropathy, which is characterized by a very early age of onset, between 10 and 24 months of age, which is quite early because usually patients start to develop clinical signs between 10 and 20 years. And it's also characterized by a quite slow progression. Also, the nerve conduction velocities are strongly reduced. Most of the time, they could not be measured in almost all patients that described to date. And there is a very peculiar characteristic for those patients. It is for those for which we have been able to do nerve biopsy. We have detected those abnormal myelinated fibers that we call myelin outfoldings, which are due to hypermyelination defects. So this shows that the disease here, it's originally hypermyelination defect that then will lead to demyelination. As you can see here in the nerve of a patient, there are almost no more fibers, and the remaining fibers, they are completely abnormal with myelin outfoldings. So this is a characteristic of this CMT subtype, and this is very important because this is the basis of most of our working hypotheses for the rest of the work and the talk. So for those who are old enough to have worked with microsatellite at the good old times of genome widescreen, we have genotyped for this space, in this family, 400 microsatellite markers. And we have realized homozygosity mapping, which consists of localizing the locus containing the gene by searching homozygous regions shared by all the affected individuals which contain the causative genes. We did this in the Lebanese family and in the Algerian family, which allowed to identify a 11.5 megabase identical by descent region at this region of chromosome 12. We had a very high lot score also for maybe the young ones, they won't know what is lot score, but it was used to have some statistic about the certainty of our localization studies. So we had this locus on chromosome 12, and we published a first paper describing CMT4H, so the CMT subtype and the localization on chromosome 12. On that time, it was still possible to publish localization, which is, of course, now completely impossible. So, and at that time, it was in 2004, 2005, the genome was sequenced for a few years, and we had not access to NGS technologies yet. So what we were doing is we were looking in the database what genes were in the locus that we had identified, and trying, based on the role of the proteins encoded by the genes, we were sequencing the genes one by one after the other, by Sanger sequencing. That's what we did here, and we sequenced the gene in red here, and finally we sequenced FGD4 and found mutation in both families in this gene. As you can see here, both mutations affect the same codon. It's codon, it's the methionine at position 298 in the protein. And in the Lebanese, we had a T2G change, which was supposed to lead to be a missense mutation, changing a methionine to an arginine. In the other family, we had a T2C change, leading to the change of a methionine to a threonine. As you can see, it was highly conserved among species, and also in the protein family from FGD1 to FGD4. Back also at this time, we were just controlling, doing like hundreds of patients, one plate, 196.8 well plate, checking that we did not find the mutation in those 100 patients. And that we considered that those mutations were the causative mutation here. We were able to demonstrate that this supposed missense mutation was actually a splicing mutation in the Lebanese family, because if you can see here, the T to G, it changes ATG to AGG, which will change the methionine to the arginine. But most importantly, it will activate this GT here, which is a cryptic donor splice site, and lead to the removal of the last 100 base pair in the transcript. And therefore, the mutation is not a missense mutation, it's a frameshift mutation, leading to complete loss of the protein. We published this second study in the American Journal of Human Genetics in 2007. Since there, several mutations have been described in the gene. Around 30 mutations now are described in the literature. So CMT4H remains a very rare subtype of CMT disease. Maybe we have something like 50 patients described in the world with mutation in its genes. Most of them are homozygous, and most of them carry mutations which are leading to loss of function, like frameshift, nonsense, or splicing mutation. So now I will go more into detail into the pathomechanisms which underlie this disease. So what is Frabin? What is the FGD4 gene? What does it encode? It encodes a protein called Frabin for FGD1 related F-actin binding protein. It's a 766 amino acid protein, ubiquitously expressed. And it has a GDP to GTP exchange activity. It's what we call a ROGEF. So it activates small GTPase such as CDC42 and RAC1. Most interestingly, this protein it has 5 functional domains. So the DH domain, responsible for the ROGEF activity, 1 N-terminal F-actin binding domain, and 3 C-terminal domain, which are involved in binding to polyphosphoinositides. Polyphosphoinositides are lipid signal messengers involved in signal transduction and membrane trafficking. Based on the function of the proteins, the fact that we had a lot of function and the multiple domains that we have in the protein, we have built several hypotheses for the pathomechanism which could lead to the peripheral nerve disease when we have mutation in this ubiquitously expressed gene. And here I won't talk about everything because again, this is a very, very long story, much of work that has been done, many paths which have led to nowhere. But what I will present here today is this hypothesis of altered phosphoinositide signaling when we have mutation in the gene and when we lose interactions with polyphosphoinositide. We make the hypothesis that considering the role of polyphosphoinositides in signaling and especially in vesicular trafficking, we have alteration of vesicular trafficking here in CMT4H. To study this hypothesis, we have taken into account that in all patients with CMT4H for whom a biopsy was made, we have observed those myelin outfoldings which correspond to hypermyelination and which are probably related to problems of vesicular trafficking in Schwann cell. Vesicular trafficking in Schwann cell is a major process which needs to be really tightly regulated. Otherwise, you can have those strongly perturbed myelination events. So to study these altered pathways, we have built and used 2 models. We have made a mouse because there was no mouse model available at the time. And what we did is that we have used a conditional knockout model for FGD4, meaning that we specifically delete the expression of FGD4 in Schwann cells by crossing flopped mice with a transgenic mice expressing the Cre recombinase under the P0 promoter. P0 is a gene which is specifically expressed in Schwann cell. It controls the expression of a myelin structure protein, which is P0 or MPZ. So we have those mice, which we name like this, you will see throughout the talk. And from those mice, we also have derived an in vitro cellular model, which allows us to do myelin in vitro. What we do is we take mouse embryo from knockout mice. We remove the spinal cord and we take the dorsal root ganglia here that you can see here. And in those dorsal root ganglia, we have the cellular body of the sensory neurons. And also we have immature Schwann cell. We put them in a culture flask. And at day 5, we induce myelination with ascorbic acid. And this allows us, as you can see here, to obtain nice myelin segments here in red after 15 days of culture. So it's a very interesting tool to study myelination events in vitro. Okay, what did we get when we studied those two models? We, in vitro, we were able to show that in vitro myelin derived from knockout mice display a large number of abnormalities, as you can see here, as compared to wild type. And these abnormalities mimic the myelin outfoldings that we observed in patients' nerves, and they are significantly increased in the knockout cultures as compared to the wild type culture. We have been able to demonstrate that the occurrence of those myelinating outfoldings are due to the loss of FGD4 because we re-expressed FGD4 in the knockout co-cultures by transducing them with a lentivirus expressing FGD4. And as you can see here, this leads to a strong diminution of the number of such myelin abnormalities in knockout co-cultures. I will try to go faster because I think I will run out of time. Okay, in vivo, we had also the same results, the presence in the sciatic nerves of the mice of those myelin outfoldings as soon as 3 months of age, and which increase with time, as you can see here. We also have a strong denervation of muscles in aged mice 1 year, which show that at the end, although we have a demyelination disease, we end up with axonal degeneration as well at the end of the process. So here we have demonstrated that the loss of Rabin is causing those myelin outfolding, but still we did not know what is the signaling pathway which is altered. And as we know, I will go fast here also, that the most important pathway to induce myelination is the neuroglin-1 type 3 ERBB2 pathway. This is a pathway which is initiating by this growth factor present at the membrane of the axon, so of the neuron, and the interaction with the receptor at the surface of the Schwann cell will induce a downstream signaling events which will induce myelination. So we studied this pathway in our models, the same models that I've shown you before, in vitro myelin models. And what we can see here is that we have strong upregulation of neuroglin-1, the receptor, the phosphorylated receptor, showing that we have upregulation of this neuroglin-1 type 3 pathway, and also of the downstream activated pathway with AKT and mTOR, which also are upregulated in conditions of loss of Rabin in the co-culture. In vivo, we obtain also the same results with upregulation of this pathway in the sciatic nerves of full knockout mice. So this shows that this pathway and dysregulation of this pathway is involved in the myelination abnormalities that we have in the mice model, in the in vitro model, and probably in the patient. So here we thought of trying to target this pathway in order to restore a normal myelination. And to do so, we used the fact that neuroglin-1, here it's a transmembrane protein in the membrane of the axon, and it has to be cleaved by two types of enzyme. This one, BAS, she has an activating, when she cleaves neuroglin-1, sorry, she will activate myelination, allowing the binding to the receptor, while when it's cleaved by TAS, it will inhibit the binding to the receptor and inhibit the myelination. That's why we choose to activate this enzyme by using a molecule which is called niacin. Niacin, it's a vitamin B3, so it's an FDA-approved drug. It's already used in clinics for the treatment of lipid disorders. So it was a good pharmacological molecule to choose if we want to go to clinics, if we show that there is some effect on the abnormal myelination. And also it was used in another model of dysmyelinating CMT4 disease. Here are the results that we obtained, again, in vitro, in vivo. By treating with niacin, here is the untreated co-culture, here's a treated co-culture, we strongly diminish the number of myelin abnormalities in knockout co-culture treating with niacin. We decreased by 60%. We also strongly downregulate the abnormality upregulated and neuroglin-1 ERBB2 pathway, so which was the aim of the process. So it shows that this is a good idea to treat those cultures with this molecule. So we went to treat for preclinical studies in vivo. We treated our mice starting at age 4 weeks before we have the first defect in the nerve, so before 3 months, and we treated every 2 days by intraperitoneal injection during 2 months. Again, we showed a strong amelioration of the histopathology of the nerves, with a strong decrease of the number of those myelin outfoldings that you can see here. It's an untreated mice. Here it's a treated mice. You see that it's almost normal. So I think here I will pass because I have no time. As a conclusion, we can say that these very, very long studies, it began, I mean, in 2002, so it's a long time ago. We showed that the specific deletion of FGD4 in Schwann cell is sufficient to induce myelination abnormalities, like the ones that we observed in nerves. And we have built a strong in vitro and in vivo model. We have shown that the aberrant myelination is accompanied by an upregulation of key factors from the neuroglin-1 type 3 ERBB2 pathway, as well as of its downstream effectors, that the loss of fibrin in Schwann cell and the modification of interaction with its partner impairs endosomal trafficking, and that most importantly, the modulation of this neuroglin-1 type 3 ERBB2 pathway using niacin, an activator or STAS, improve myelination in vitro and in vivo, which strongly pushes us to go maybe further to maybe go into for clinical trials if we can perform those two additional trials in the mice. And also we are intending to screen for other molecule. Okay, I try to stick to the time, so I want to thank some people, of course, my team, and my dear collaborators from Lebanon, Professor Megarbane, Eliane Choueri, and Nabiha Salem, who participated in sampling the patients, and the whole team from the ex-lab at Saint Joseph University, Professor Gérard Lefranc, who is a French, also a professor who induced me to go to Lebanon. And also I want to special thanks to those two girls. So this is Nathalie Bernard-Marissal, she's a postdoc, she did most of the niacin and in vivo work. And Lara El-Bazal, she was my Lebanese PhD student, and she did all the in vitro work. So they have put much effort in this work, and also people from our lab, from the animal facility. And I will finish with nice photos from my city. And I thank you for your attention.